Hello, class. So I am out today, so I have left you this video in hopes that it will help you with learning the material while I'm not here and also helping you with working on the lab that I have assigned for today's class. So taking a look at our outline for today, 10/31/19, Halloween, we're still on the unit of photosynthesis. We're going to be talking about the topic of photosynthesis today as well, specifically factors that affect the efficiency of photosynthesis. So for classwork, you're going to start off by watching this current video that you're watching right now, whereby I'm going to give you guys a little bit of notes on the factors that affect photosynthesis, and I'm going to explain how you guys can do the lab. Once I'm done explaining all of that, it'll be up to you guys to actually work on the lab for factors affecting photosynthesis, and I will walk you through how to do that. And that is what we are doing in class today. In terms of homework, the lab you are working on today will be due 1 week from today, so next week, Thursday. So make sure you guys finish that up. Other than that, you have some homework. There is an ed puzzle for Chapter 8.3, a Quizlet for Chapter 8.3, and chapter reading from the textbook for Chapter 8.3. I assigned this to you guys last class after your test. Some of you may have done it already. If you have not yet done this already, if you are in my honors class, this assignment is due on Friday, the 1st of November. If you are in my CP class, this assignment will be due on Monday, the 4th of November. Our objective for today is the same as it has been for the last couple of days. We need to learn how to use a model to illustrate how photosynthesis uses light energy to transform water and carbon dioxide into oxygen and chemical energy stored in the bonds of sugars and other carbohydrates. All right, so to start off with, let's begin going over the notes and explanation of the lab. So in terms of factors that affect photosynthesis, there are several of these factors. The first that we're going to talk about today is temperature. So temperature can affect how efficient photosynthesis is able to run. If the temperature is too low, photosynthesis might not work correctly, and if the temperature is too high, photosynthesis might not work correctly. And the reason for that is because photosynthesis depends on enzymes to be able to do a lot of the chemical reactions that take place in the process of photosynthesis. So you may remember from the beginning of the year that all enzymes have a temperature that they work in best. The enzymes related with photosynthesis work best between a temperature range of 0 degrees Celsius, so above freezing, up to 35 degrees Celsius. If you remember, the reason why temperatures above 35 degrees Celsius might be inefficient for the enzyme is because the enzyme begins to denature, which means that it begins to change shape. The active sites for the enzyme no longer fits the substrates which it acts upon, and it's not going to work as well. So those changes can be brought upon by too much heat, aka over 35 degrees Celsius, or too high of a pH level or too low of a pH level, too acidic or too basic. However, for right now, we're focusing on temperature. So enzymes for photosynthesis work best between 0 degrees Celsius and 35 degrees Celsius because those are the temperature ranges in which their enzymes do not denature and they're able to work efficient enough to get the job done. Another factor that affects how well photosynthesis is going to be able to operate is light intensity. So as you increase the amount of light, this will also increase the amount of photosynthesis that is occurring. However, only up to a maximum threshold. If we take a look at this graph over here, we can see that if this is representing on the y-axis is representing our rate of photosynthesis and our x-axis is representing an increasing light intensity, you can see that as we increase the light intensity, the rate of photosynthesis increases quite dramatically up until a certain point at which it plateaus and levels off. The reason why this plateau and leveling off happens is because light has become saturated. There's only so much light that the plant can absorb. So if there's no light, photosynthesis isn't going to be working very well. If you increase the amount of light, the plant is going to absorb more and more light, undergo more and more photosynthesis, until you're giving the plant so much light that it really can't absorb anymore, and the benefits of increasing light intensity taper off and the graph will plateau. So, in general, increasing light intensity increases the rate of photosynthesis up until a certain point at which it plateaus. Another factor that affects the efficiency of photosynthesis is water. As you all know, you need to water plants in order for them to survive and grow healthily. And part of that is because water is one of the important reactants for the process of photosynthesis. If you do not supply the plants with enough water, so if there's not enough water, the process of photosynthesis is going to slow down or stop entirely. If you give the plant too much water, that's going to kill the plant, and that's also going to stop photosynthesis. So you want to find a happy medium between no water and too much water, or too little water and too much water. So you want to water your plants as they need, and that's the relationship that water will have upon the efficiency of photosynthesis. Another factor that affects the efficiency of photosynthesis is light color. We talked about the efficiency of photosynthesis and how it's related to the intensity of light. However, it's not just related to the intensity, it's also related to the color of that light. So photosynthesis is going to work best with violet/blue colored light and red/orange colored light. The reason for this is because if we look at the different pigments that are involved in photosynthesis, we can see that they mostly absorb colors of light in the violet, blue, orange, and red parts of the spectrum. So photosynthesis will be least efficient with greenish yellow light because the pigments do not absorb the energy from green or yellow light. Last one of the factors we need to cover in regards to the effect that they have on photosynthesis is carbon dioxide concentration. As we know, one of the reactants of photosynthesis is carbon dioxide gas, which plants absorb through their leaves. If there is not enough carbon dioxide present, the plants aren't going to be able to photosynthesize efficiently. However, as we increase the carbon dioxide concentration, this will lead to increased photosynthetic efficiency. However, the question is to what extent? It's most likely one of these graphs similar to light intensity where it plateaus off, because as you increase the carbon dioxide concentration, there's more carbon dioxide available for the plant, so it's going to work better. However, at a certain point, the plant can't absorb any more carbon dioxide than it already is. So it will increase the rate of photosynthesis until it becomes saturated with carbon dioxide, at which point in time, any increase in the amount of carbon dioxide will not increase the efficiency of photosynthesis. It will just taper off and become a plateau at the end. No matter how much higher you make the CO2 concentration, the rates of photosynthesis will plateau at around that point. So those are the factors that affect photosynthesis. To summarize, basically, carbon dioxide. If you increase carbon dioxide, you increase photosynthesis up until a certain point at which it plateaus. Colored light. Photosynthesis is going to work best with blue, purple, orange, and red light. It is not going to work well with green or yellow light. Water. You don't want to underwater your plants because that will slow down or stop photosynthesis. You also don't want to overwater your plants because that could kill the plant and stop photosynthesis. You want to find a happy medium of just giving the plant enough water that it needs, but not overwatering it or underwatering it. In terms of light intensity, increases in light intensity increase the rate of photosynthesis up until the light saturation point, at which it will not increase the rate of photosynthesis anymore. It will just plateau. And lastly, temperature. Remember that the enzymes that work in photosynthesis need to be between the temperatures of 0 degrees Celsius and 35 degrees Celsius in order to operate correctly. Okay. So that covers off the notes of what the different factors are that affect the efficiency of photosynthesis. Now let's take a look at the lab you guys are going to be doing. So if you go to Google Classroom and you navigate to the classwork page, you should find under today's date, I have not posted it yet over here, but it'll be under the date of 10/31/19. You'll see a link that says factors affecting the efficiency of photosynthesis lab. If you click on that link, you will be brought to a document that looks somewhat like this. First thing you're going to want to do is go File, Make a Copy, so that you have a copy of this lab that you and your lab group can work on. Now, you are allowed to work in groups on this lab, groups of up to 4 people per group, no more than 4 people per group. You can work in groups of less than 4, but no more than 4 per group. All right. So now that we've made a copy, we have this little neat template that I made for you guys. The title of the lab is already in here. All you have to do on this first page is insert your names of whoever was in your group over here. Make sure to put the main names of all of your group members so that everyone gets credit. And then you could insert the class name, which would be biology, my name, Mr. Markram, and the date, which will be 10/31/19. Scrolling down to the next part of the lab, it says purpose. The purpose of this lab is to investigate how different factors affect the efficiency of photosynthesis, something we already talked about at the start of class through my video. So we need to create 4 separate hypotheses for how the 4 different factors of temperature, light intensity, light color, and carbon dioxide concentration will affect the efficiency of photosynthesis. So fill in the blanks over here. So if the effect of light intensity on the efficiency of photosynthesis is studied, high or low light intensity will be the most efficient. So fill in this blank with either high or low. If the effect of light color on the efficiency of photosynthesis is studied, blank colored light will be the most efficient. So you can pick any color there. It could be blue, it could be violet, it could be red, it could be orange. So pick one of those colors to make your hypothesis for. If the effect of temperature on the efficiency of photosynthesis is studied, high temperatures, low temperatures, or medium temperatures will be the most efficient. And lastly, if the effect of dissolved carbon dioxide concentration on the efficiency of photosynthesis is studied, high or low dissolved carbon dioxide concentration will be the most efficient. So fill in your predictions for those different hypotheses. Then you are going to start the procedure. So I've outlined this entire procedure for you guys over here. So if you can't remember everything I say in the video, all of this information is still available to you to consult in the procedure for the lab. First thing you're going to need to do is click on this link here that says open the following link to the virtual lab. So if we click on that and we click on this link, we will be brought to the following page. So as we look at this page, you can see that it's saying Adobe Flash Player is currently blocked. So what we need to do is scroll up here to the top of the page where it says not secure. Click on that. On mine, I can change the setting right up here, but I know sometimes your Chromebooks are a little bit difficult, and I checked on the computers on the side of the room. So if your Chromebooks are not working, you're welcome to use one of the computers on the sides of the room, one of the desktops, and they should operate. I tried this on them earlier, and it worked perfectly fine for me. So you can either allow it over here if it gives you the option. If it's not giving you that option, if it is not giving you that option, you need to click here where it says not secure. Go down to where it says site settings. Scroll down to where it says flash, and you want to change this block default to allow. Now that you have changed these permissions, you can now close out of the settings tab. And it should say here to apply your updated settings to the site, reload the page. So I will reload this page. And as you can see, I now have the following in front of me. So here's where you're going to change all the different settings for the different factors affecting photosynthesis. So if we take a look at the procedure, first factor we're going to study is light intensity. So in order to test the effect of low light intensity, set light intensity to 10, light color to white, temperature to 25 degrees Celsius, and dissolved carbon dioxide to low. So you would scroll over here, you would set the light intensity to 10. So this light bulb over here, we want to scroll and change that to 10. We also want to change the color of the light by clicking on this button over here to change the color. So we want white, which looks kind of gray in this instance. So you want to have that gray looking color. We want to change the temperature to 25 degrees Celsius. So temperature, we want to bring up to 25 degrees Celsius. And carbon dioxide concentration. Right now, this is low carbon dioxide concentration. You can tell because you can see this line up here is near the top of the bottle. That means we've put none of the carbon dioxide inside of here. So this is low carbon dioxide concentration. So that's how we want to leave it. We do not want to click on this bottle to change anything. So here are our parameters. Now, what you are going to do is once you've set all those parameters, you want to hit the green button on the timer to begin your data collection. Once you have begun the timer, you want to count the number of oxygen bubbles produced by the plant over a 1-minute period. You want to end the collection when the timer reaches 1 minute by hitting the red button and record the number of oxygen bubbles produced in the appropriate section of the data table below. So that would be right over here. Looks like I need to get rid of some of that data. So over here on the data table, let me just erase these real quick. So on the data table where it says low light intensity, you are going to come over here. You're going to hit this green button. You're going to count how many bubbles over the course of 1 minute. So that was 1, 2, 3, 4, etc., etc. And you want to wait until it's been a minute, and then you hit the red button, and then you record the number of bubbles that you got over here in this column on your data table for low light intensity. Then what you are going to want to do is you're going to want to change it to medium light intensity. So you want to change the light intensity to 30, keep the light color white, keep the temperature 25, keep the dissolved carbon dioxide low. So the only thing you're going to change now is changing the light intensity to 30. So we move this up to 30, and then we can go ahead, hit the green button. So let's hit the red button, hit the green button to get started with. Hang on, red button, red button. Okay, so if we just hit the red button, it seems that okay, the green button stops it, the red button just resets it. Okay, so we hit the green button to start timing. So 1, 2, 3, 4, 5, etc. for a total of 60 seconds. You then stop the timer. You can record the number of bubbles over here for medium light intensity, and then you'll move on to do the same thing for high light intensity. So you'd want to go in here, change the light intensity to, let's see, what is the value I say here? Change the light intensity to 50. So you want to come in here, change the light intensity to 50, and then you want to reset your timer, start timing, count the number of bubbles, 1, 2, 3, etc., until you've done that for 60 seconds, stop the timer, reset the timer, record your data in the data table box right over here. Then you want to do some experiments for the color of light. So if we take a look at what we're doing here, we want to set the light intensity to 30 for all of the trials, temperature to 25 for all of the trials, and dissolved carbon dioxide to low for all of the trials. We then want to change the color of light to orange to start with, and then we'll record our data. So we want to have our light intensity back down at 25, our temperature at 25, and our carbon dioxide at low. Actually, it looks like light intensity is 30, so let's move that up to 30. There we go. Okay. Now we want to change the color of light by hitting this button, making it orange light. We then can go ahead and we can time over 1 minute. So we hit the timer, 1 minute. So you count the number of bubbles, 1, 2, 3, etc. You're going to record that once you're done with a minute, reset the timer, record your data over here where it says orange colored light. So you're going to fill in the number of bubbles that you recorded in a minute under orange colored light. You're then going to come back here. You want to change it to a different color light. We're going to do green colored light next. So leave all the other variables the same. All you need to change is the color to green. Record how many bubbles get recorded within a 1 minute period. There's 1, etc. You want to time for 1 minute, and then you can record the data over here under the box that says green colored light. Lastly, for blue colored light, you hit the button again to get blue colored light, to leave all the other parameters the same, and then you want to time for 1 minute how many bubbles, stop your timer, record the number of bubbles over 1 minute. Last one of our trials that we're going to be doing here. Oh, actually, we have two more, carbon dioxide and temperature. So for carbon dioxide, there's only two options, low and high carbon dioxide. So looking at the procedure over here, we are going to set light intensity to 30, light color to white, and temperature to 25, and we're going to keep carbon dioxide low to begin with. So we want to set this to white, light intensity to 30, temperature to 25, and we want to leave this bottle looking like it's full of this liquid, so it's at low. Then we want to reset our timer and time how many bubbles occur within a minute. Record that value for the low carbon dioxide condition over here. Next, you want to record the number of bubbles for the high carbon dioxide condition. So for this, you leave everything the same. The only thing you're going to change is hit this bottle, and as you can see, the bottle is now half empty. That means we've put a bunch of this carbon dioxide in here with the plant. So it's now a high carbon dioxide condition. We want to reset our timer. And we want to begin timing for 1 minute, counting the bubbles. 3, 4, 5, etc. for 1 whole minute. Then you reset the timer. So that should give us our data values for low and high dissolved carbon dioxide concentration. Last one is we have a low temperature, medium temperature, and high temperature condition. So what we would need to do here is, looking at our procedure, we want to set light intensity to 30, light color to white, temperature to 10, and dissolved carbon dioxide to low. So we want to have light white, temperature should be down at 10, carbon dioxide should be back to low concentration, so it needs to look full, and everything here is set. White light, light intensity 30, low carbon dioxide concentration, and we want to set our temperature not to 40, to 10. There we go. And we want to start our timer, record the number of bubbles produced in 1 minute under this condition. Then what you're going to do is you're going to keep all the other variables the same in terms of light intensity, light color, and dissolved carbon dioxide. The only thing you're going to change is the temperature variable. So you would change the temperature from 10 to 25. And then you will reset your timer, count how many bubbles produced in 1 minute at 25 degrees Celsius, stop the timer, record that data in your data table under the medium temperature condition. And then lastly, high temperature condition, we are going to set our temperature to 40 degrees Celsius. So keep everything else the same. Click on the thermometer, bring it up to 40. Time how many bubbles will be produced in 1 minute under the 40 degrees Celsius condition. Stop the timer, record that data over here in your data table, and you should be all set. Other than that, once you're done collecting your data, there is a Google Sheet to go along with this lab. Let me actually go ahead and highlight that in blue for you guys so that you know that is a hyperlink for you to click on. So highlighting this, making it blue so it stands out as a hyperlink for you. So if you click on that Google Sheet, as it says, insert the 4 graphs. So if you open this up, you will get the following data table. What you are going to need to do is you are going to need to wait for it to load first off. Seems to be taking a little while for me. There we go. It is almost done. And now we have our data table. What you need to do now is go File and click Make a Copy. Then hit OK. This will open up your own editable copy of the lab data table. And now you can go ahead and plug in the data you just recorded throughout the lab in these data tables. What should happen as you plug in that data is these graphs should automatically create themselves for you guys. I then want you to click on these graphs, click on these 3 dots over here, copy the graph, go back into your lab report, and paste each one of those graphs under the appropriate heading. You can just say paste unlinked and hit paste. And there is my graph for the light intensities and how they affect the rate of photosynthesis. Do the same thing for all of those 4 different graphs. Write a conclusion. Here is what you need to include in that for your summary. Make sure that you include a summary of why we did the lab, how it relates to what we're doing in class. Think about the big picture idea. Your analysis of data. You're going to have 4 different hypotheses here. So you want to state all 4 hypotheses, state out of those 4 which were supported, and provide evidence from your data saying why it was or was not supported. So you've got quite a long analysis of data there because you need to take care of all 4 separate hypotheses. Lastly, discussion of error. Think about any factors that might have affected your results, things like miscounting your bubbles, things like this being a virtual simulation. It might not be indicative of reality, so the reliability of the simulation can be called into question, or any other factors that you think may have affected the results on your lab. Lastly, you can use the notes provided in the beginning of the lesson today that are found under Google Classroom under today's class notes, as well as your own problem solving skills in order to answer the following questions in your own words. So answer these 5 questions, and then you can turn this lab in. Remember that this lab is going to be due next lab period, which will be on the 7th of November. That is what you are doing in class time today. I hope you guys do well. Be good for whoever is covering my class, and I hope to see you all next class time. Let me know if you have any questions by sending me an email over the weekend. Thank you. Enjoy your class.